Hello, everybody. I'm Sharon Hillier, and welcome to the second press conference for the annual conference on retroviruses and opportunistic infections, known as CROI 2021. We ask in your reporting that you do refer to the meeting as the Conference on Retroviruses and Opportunistic Infections, or CROI. And we today have a really exciting press program featuring several highlights from today's scientific program. And as you know, these are just a few of the many excellent presentations we've had today, including the Science Spotlight presentations, which I hope you've had a chance to visit. Each of today's speakers will give a really short overview of the studies, no more than 2 to 3 minutes. It up for live Q&A. Please put your questions in the Q&A feature. And please, when you list your questions, list the outlet or the media group you're representing so that we can identify that correctly. If you would like, you can put your question in the Q&A box, or you can just list your name and your media affiliation and just write question, and then we'll unmute you and you can ask the question yourself. So 2 options: either put the question in the Q&A box or write your name and your affiliation and write question, and we'll unmute you. We may not be able to get to all the questions today, but we'll allow as many as possible, and apologies in advance if we don't get to yours. The media team will help you by circulating a recording of the press conference within a few hours of our completion today. I want to thank all the speakers for joining us and all of you journalists for attending this press conference. And then I'd like to introduce my co-chair for the press conference, Rick Kaup.
Hi, thank you everyone for attending. We have I think a really wonderful lineup today. As you can probably see, the first half of the press conference will be dedicated to COVID, or SARS-CoV-2, and then the second half to HIV. And the way we'll do this is the first talk will be on COVID vaccination, and we'll have that presentation and questions, and we'll follow that up with 3 presentations on monoclonal antibodies for prophylaxis and treatment. And then we'll have all 3 of those, and then we'll take questions for those together, because there are going to be many questions I'm sure that you will have that are very similar for those 3 presentations. So let's start this off with COVID vaccination, mRNA vaccine in SARS-CoV-2 experienced people, from Mark Mulligan from New York University. So Mark, can you give us a brief overview of your findings?
Sure. Thank you very much, Dr. Kaup, and thanks to the organizers for giving us this opportunity. You know, for a vaccinologist, it's been gratifying to participate in clinical trials. We've done that now for at least 3 different COVID vaccines at our institution. But now that there are EUA vaccines being used, 60 million as of today roughly, it gives us a chance to do investigator-initiated research. And one of the things we were quite interested to do was to compare the response in people that had COVID back in March and April, for example, and then get vaccinated, versus those who've never experienced COVID and get vaccinated. And we've done that in an exploratory study and reported those results at this meeting. So what does it all mean? I assume that you all are familiar with the data, so I won't go back through it. You know, what I think we've done is a hypothesis generating study. And the hypothesis that I think our data supports is that people with a history of COVID might only need 1 COVID vaccine, 1 dose of an mRNA COVID vaccine. But we can't make that recommendation. I'm not making that clinical recommendation, but I do think it's something worth paying attention to. What we showed in the study is that the magnitude, the amount of antibody, the quality of the antibody, the B cells, the ability to neutralize the variants, all of those after 1 dose in people who are experienced is very similar. People who've had COVID before is very similar to what we see after 2 doses in people that have not had COVID before. There are many limitations to our study, including that everyone did get 2 doses eventually, so we can't follow people over time. to see how one dose versus two dose compares. But it's actually useful information, and it does make one important observation as well, and that is that in many people, having had COVID before really provides strong preparation or priming to receive a vaccine and have a very strong response, even to one dose. So I think this is an interesting area that deserves more study. And I'll stop there, Dr. Kalp.
Great. Thank you. So I think what you're doing is very important, and I really appreciate the fact that you shared these data so early. So we do have a first question from Liz Heileman from PAUSE, and she asks, I've been seeing suggestions that people with long COVID are experiencing improvement after vaccination. So were you able to look at this, or do you have any insights?
Yeah, no, unfortunately I don't, Liz. We enrolled nobody with long COVID. All of our, we did this as quickly as we could, and so it was the healthcare workers from our university who got vaccinated, and that's who we enrolled. A lot of them had COVID, but none of them had long COVID, so our study does not inform on that at all. In general, antibodies, monoclonals, which we're going to hear a lot about in a minute, for treatment are useful in an early phase, outpatient phase. There's not a lot of data about their possible use or about vaccine as a therapeutic vaccine, which is what that would have to be. So I'd be very interested to learn more about that myself.
Thank you. We have a question from Lynn Peterson from Trends in Medicine. I think you want to ask that yourself. So Mark, if we can unmute her.
Thank you, Dr. Mulligan. Hi. So how long did people have COVID-19 before you gave them this vaccine? Because if it was relatively new, that would be different than if they had had it. And we don't know how long the protection lasts from getting COVID-19.
Yeah, that's a very important question, Lynn. We know that the interval between a prime and a boost, which is sort of what we're talking about, the infection, the COVID-19 being the priming event, and then the first dose of vaccine being the boost, is very important in vaccinology. And in general, the longer, the better. We, in the data we reported today, we had 13 COVID-experienced people. 2 of them had been asymptomatic, so we don't know when they were infected. The other 11, 9 were infected back in March and April, so an average of 8.4 months since their infection, and then 2 were infected in December, so very recent. The numbers are just not large enough yet to let us discriminate whether that interval matters, but the very strong responses we saw included people both with infection 2 months ago and with people 8 or 9 months ago.
Great, thanks. We actually have quite a few questions. I'm going to try and go through some of these quickly. So Claire Wilson from New Scientist, did you see similar results with T-cell activity?
We didn't present our T-cells in this, mainly because of time, but it's something we're working on, and I don't think I can say anything more about that yet. We're still working on it.
All right. From Jeff Barry from Positively Aware, there may be people who only end up receiving 1 dose of an mRNA vaccine for various reasons. Could a registry or observational study be established to follow those individuals?
Well, I love the way you're thinking. We're doing that informally. I have about 10 who, this was reported in the New York Times, and we started getting phone calls. And I saw one yesterday, and she said, I'm terrified to get my second dose. I had a really tough experience. And I said, well, if you're not going to get it, or if you are, we'd love to follow you over time, and you would be making a contribution. So there are those people out there, and I think we should do that. Absolutely.
And that leads right into a good follow-up question from Ed Sussman from MedPage Today. What kinds of reactions to the vaccines occur in people who have experienced COVID? Are they the same types of reactions that people see with their second vaccination?
Yeah, the reactions we see with the second are the same as with the first in our phase 3 trials of RNA vaccines. there is some difference in their intensity, a little bit more frequent and maybe a little more significant reactions, all still within the range of tolerability, acceptability, and well worth it for the protection you get. With the experienced people in our study, we don't have, we haven't done that readout yet to see if they differ in any way. But my gut feeling is that they're the same types of reactions, and I don't know yet if they're more intense with prior infection compared to no prior infection with first dose. That's an interesting question. One group at least has reported they believe that there are stronger reactions in people, it's suggestive at least, in people with prior COVID.
And then we have a question from Amr Malik from the News Pakistan. How can the real prevalence of COVID-19 be determined when the testing rate is so low?
Yeah, I think that question has to do with something I'd like to say, and that is that, you know, the most important job we have now is to get everybody vaccinated and to follow the guidelines of 2 doses for RNA vaccines. Of course, now 1 dose is an option with J&J, and I don't recommend that we change that. In the U.S., vaccine is becoming more widely available. For the whole world, our study, you know, could be informative, but we have to do more work to know if it really is something that would be worth trying to layer into a massive vaccine campaign. When you do this kind of huge public health undertaking, it's got to be simple, protocol driven, easy peasy. And I worry a little that, you know, as you say, in places where there's not even any testing, you know, how could you ever implement wisely something like this? But that's not to say that there might not be ways to get at this, but it shouldn't, we need to know more first.
Yeah. And then finally, one last question, a follow-up question from Claire Wilson. Is there any evidence from phase 3 trials in seropositive or previously experienced individuals?
There is, Claire, in a limited way. People with a known diagnosis were excluded, but we didn't do a screening blood test. So there were people, several hundred, who were in the trials and were vaccinated, and they had the same, you know, the preliminary reporting from the EUA packages. They had the same safety and efficacy profiles. I have not yet seen a report, and I've asked some of the companies this, and they said they haven't done the analysis yet on their antibody responses, if they got the same thing we got. So good question, and the data are probably there. It just hasn't, to my knowledge, been analyzed yet.
Yes, I do believe that certainly for the Moderna trial, somewhere between 10 and 20% of people who got vaccinated in retrospect were seropositive at the time of vaccination. So I think those data will fall out of those trials.
Yeah.
Great. I think we have to move on now. As I said, we're going to do 3. So thank you, Dr. Mulligan. That was fantastic. We're now going to do 3 monoclonal antibody presentations. And then we'll take questions together at the end of that. So the first is from Mike Cohen. It's on bamlavinimab and its use in nursing homes. So Mike, please give us a nice quick overview.
Thanks. Thanks for inviting me to participate. Today, whatever time it is for you, I had the opportunity to present the results of a randomized controlled trial that represents a collaboration between NIH and the Eli Lilly company to explore bamlavinimab for prevention of SARS CoV-2 and prevention of progression of disease in skilled nursing facilities. And so in this particular trial, both the residents of the facility as well as the staff were invited to participate. And probably one of the more remarkable parts of the trial is that Eli Lilly outfitted about 10 vans that crisscrossed the United States when a facility found themselves under COVID attack, and would try and arrive when an index case or a few infected people had surfaced. All of this started before vaccines, all of the planning and thought started before vaccines were available. So this kind of passive immunity was very provocative in terms of how vaccines might work and how much prevention could we observe. For this particular talk, we focused primarily on about 1,000 people who were enrolled in about greater than 100 facilities. And among those 1,000 people, we focused on 300 who were residents of the facility because they're older and at much greater risk for progression of disease. The median age of those participants was 75 and 76, so it's placebo-controlled. In this trial, among the residents, about a quarter of those who volunteered acquired SARS-CoV-2 infection, and some progressed to SARS-CoV-2 disease. And in those who received 4,200 milligrams of bamlavinimab within 7 days of presumed exposure, there was an 80% reduction in the progression of SARS-CoV-2 and a 72% reduction in actual acquisition of SARS-CoV-2. There were 4 deaths in the placebo group and no deaths in the group provided bamlavinimab. We also looked very carefully at nasal swabs to understand what would happen if you acquired SARS-CoV-2 in the placebo group or in the bamlavinimab group, and we saw quite a significant reduction. So at the first time a PCR turned positive in someone who was negative at baseline, at seronegative and PCR negative baseline, at the first moment a nasal swab turned positive, there was a very significant reduction in the concentration of SARS-CoV-2 recovered from the nose by qRT-PCR. So in summary, this particular trial, we think, is it's kind of a proof of concept, and it's provocative in the sense that it demonstrates that the idea that a passive immunity can prevent infection itself and can certainly help to prevent progression disease, we think was demonstrated by the data that was collected. Of course, this is a single antibody, and it was really one of the first antibodies out of the gate. I'd like to give credit to the Vaccine Research Center, who helped to develop this antibody. Many other antibodies have surfaced, recognizing the evolution of SARS-CoV-2 towards variants that are more difficult to control and that might require cocktails, as we'll hear about in a second. But we found the results to be provocative and compelling. Thanks.
Thank you, Mike. Okay. Next will be Michael Dougan talking about combination therapy with bamlavinimab and etesevimab. And this is in treatment of high-risk ambulatory patients. So, Michael, take it away.
Thank you. And again, I want to thank the organizers for inviting me here. And so the trial that I'm going to talk about, or that I talked about earlier today, is really building off of what Mike Cohen just talked about. So this is a combination of bamlavinimab and a second similar monoclonal antibody, etesevimab. And the real difference is, instead of it being prophylaxis, these were all patients who were infected with SARS-CoV-2, and it's a treatment trial. On average, people were getting this treatment after 4 days of symptom onset. And it's given by an infusion. Most people were getting this over an hour-long period. And the results really were very similar in magnitude to what we saw in the prevention trial. So there was a 70% reduction in the risk that people were hospitalized with SARS-CoV-2. And we saw a reduction in deaths. So there were 10 total deaths in the placebo arm and no deaths in the treatment arm. So this, again, similar to what we saw with the prevention trial, this really is evidence that giving monoclonal antibodies, in this case a cocktail, can prevent COVID-19 from becoming an early infection, progressing into a serious and life-threatening one. We can actually prevent hospitalizations and save lives. I think this is really extraordinary finding overall, and I think really is a powerful new treatment for this disease.
Great. Thank you, Michael. So the last presentation is also on combination therapy from Megan O'Brien from Regeneron. And this is the casirivimab and imdevimab antibody cocktail, better known as REGEN-COV. So, Megan?
Hey, thank you so much for inviting me to share our study. So this REGEN-COV, it's a cocktail of 2 neutralizing monoclonal antibodies, and they bind to different regions of the SARS-CoV-2 spike protein. and it's being tested for both treatment and prevention. So this study I'm going to talk to you about is a prevention study, not a treatment study. So Regeneron, in partnership with COVID Prevention Network, NIAID, and Roche, we're performing a phase 3 clinical trial to test whether an injection, a subcutaneous injection, of the two antibodies could reduce infection in close contacts of household members with COVID-19. So similar, I guess, for this audience to HIV post-exposure prophylaxis, the idea is that when you live with somebody with COVID, you are at a much higher risk to get COVID. So if you yourself are asymptomatic, you don't have COVID, no signs or symptoms of COVID, you can receive an injection of drug versus placebo in this study in the case, and it could prevent you from getting infected. So very similar to HIV post-exposure prophylaxis, but this is really COVID post-exposure prophylaxis. So similar to HIV post-exposure prophylaxis, where there's a 36-hour window where you need to receive the drug for efficacy. For this study, there's a 96-hour window. So the subject had to be enrolled and receive drug within 96 hours of their index case, the person that they're living with COVID, getting tested. So that 96 hours is important. So, we did an interim analysis of this ongoing study. So this is just a snapshot. And what we saw was that a single dose of the injection of the RegenCov cocktail prevented symptomatic infection by 100%. It prevented infection with a high viral load, meaning 10 log to the 4 copies or above, in 100%. And it reduced overall infection by 50% and decreased viral load and duration of viral detection. So really what we saw was, if you got drug and you did get infected, on average you had infection for a week, you were never symptomatic, and the viral levels were very, very low. So we don't know if that would be replication competent virus, for example. So, we really do look forward to, as everyone does, a more universally vaccinated public. We do think that RegenCov could be a critical piece of the COVID-19 therapy landscape because it provides an acute, fast-acting option for prevention of close contacts. And the virus drives the pandemic, and so we do think that antibodies for prevention of close contacts and used for early treatment, it could very well, again, in the HIV landscape, wording reduce community viral load and reduce transmission on top of vaccines being rolled out.
Great. So thank you. I want to thank all three of you for not only presenting these data, but just the great work you've done in evaluating these monoclonal antibodies. So the first question we have is from Heather Barner from Medscape, and it's to Michael Dougan and Megan O'Brien. So it's, have you or do you plan to submit to the FDA for EUA use of these antibodies for prevention?
I can't speak to the company's plans on prevention. Obviously for treatment we have.
And Megan?
Yeah, so we do have an EUA for outpatient treatment currently, and we are in discussions with FDA for the same for the prevention. Again, this is an interim look. It was not the final study results, but we are, because of the pandemic, trying to move things along quickly. So we are in discussions with the FDA about EUA.
All right. And Mike Cohen, do you know at all?
Yeah. No, and again, we can't speak for Lilly. I can speak for the COVID Prevention Network, but certainly Lilly is in discussion with the FDA about the use of their product for prevention, and probably more likely the combination at the doses that Michael described. I would think that would be the likely discussion.
Okay, so we actually have a lot of questions from Damien Coulombe from Le Quotidien de Médecine. What kind of study should be done in order to validate the use of monoclonal antibodies as a valid prophylactic treatment? And if so, what might be the targeted populations? So Megan, maybe you can start with that.
I think the studies that are being done now, these are phase 3 studies, and they're essentially post-exposure prophylaxis studies. So I think if a number of companies are showing in phase 3 studies that you can, prevent infection in those that receive the drug, that's I think quite validating for that approach. In terms of pre-exposure prophylaxis studies, in the setting of vaccine, those seem less relevant, and also those are very, very large studies that need to be done. That might be a little bit harder. Certainly, we're hearing through our compassionate use team that there's definitely patients who may not have an immune system that will respond to the vaccine. So there may be targeted populations that would need pre-exposure prophylaxis. And, I think our group and others are trying to understand how best to look into those populations.
Great. We have another question from Heather Berner. Oops. Can Mike Cohen and Megan O'Brien speak to the logistics of rolling out sub-Q injections or infusions to healthy people?
Do you want to go first, Megan, or do you want me?
You can go first, yeah, thanks.
Well, I think that by way of declaration of conflict of interest, of course, we're involved with Regeneron as well, and so this is a topic of great discussion. We find that first of all, IV infusions are very well tolerated. In other work we did, we gave like 44,000 monoclonal antibody infusions around the world that was reported. I guess some of that was discussed at this meeting as well. So IV infusions are tolerated. It's just that physicians, that the logistics are complicated, and to roll out IV infusions is going to take healthcare providers, both for treatment and for prevention, to kind of get their heads around, well, this is what I might have to do as an outpatient. But it's a single infusion, generally speaking. For sub-Q, Regeneron's been doing both IV and sub-Q, and so I'm going to let Megan take over, since you're doing both modes of delivery. You take over, Megan.
Yeah, this is really a team effort, and we've teamed up with Roche to ramp up manufacturing, and the healthcare system was really set up, I think, to provide outpatient infusions, really more for cancer patients. So I think, it really requires hospital systems and systems to really just, optimize their delivery in the outpatient setting for IV. Similarly for sub-Q, it's just a matter of, increasing manufacturing with support of other partners and getting it to the clinics. Any clinician can give a subcutaneous injection. Similar to IV, it's very well tolerated. These are antibodies directed against, an exogenous target. So we're seeing, quite good tolerability. So it really just is a matter of everybody, getting together, the systems, the companies, to really ramp this up and get it to people to reduce what I said, sort of community viral load, that this will prevent people themselves from getting sick, themselves from progressing, and also reduce sort of the viral load in the community to reduce transmission.
Thank you. And we have a question from Lynn Peterson from Trends in Medicine, and it's for Dr. Megan O'Brien. So this has to do with whether the study was really large enough to draw conclusions based upon the fact that even within households, transmission can be fairly low, about 10% of household contact. So the question is, was your study large enough to draw these conclusions?
That's a great question. It's interesting. When we started the study, the literature about transmission in households was mostly coming from China. And since then, there's been a lot of studies. And when you look at these different household contact studies, the range of transmission in homes is from the low digits percentage up to 90%. So really, depending on how you do the study, the percentages look really different. The 10% transmission, the 10% infection rate in our study just meant we needed sort of a bigger study. If it was a 50% transmission rate, we would have, answered the question with a smaller study. So part of the reason why we did the interim look was to test our assumptions and try to understand, how big does the study need to get to answer the question. But I think the results of the trial would be applicable to a household that has, a higher infection rate.
Megan, how many subjects are actually enrolled? I think, could you clarify the number of subjects actually enrolled in the trial?
Sure. Yeah. So this interim look that we did, there was 409 uninfected participants that were antibody negative, so that, had no evidence of prior infection. That's who we looked at in this interim look. These people will not be used in the final analysis because we've unblinded them, but they're still being followed for safety. But the overall study, we've enrolled 3,300. So we have, a little over 2,500 to look at. So that's a much bigger data set, 2,500 versus, 400. So we have to look at the overall study, but we're hopeful that the trends will stay the same. But it's a very important point that this was an interim look and this is not the total study results.
All right, I think we have time for one last question. This is to all three of you. How long do the infusions take for your monoclonal antibodies? And this is from Heather Borner. So Mike Cohen, then Michael Dougan, and then Megan O'Brien.
Yeah, the infusions, I think, it depends on which cocktail, but I believe most are coming down to about 20 minutes, 21. And it's funny because one of the goals is to make these available in pharmacies, which have minute clinics. And so as you have this discussion with pharmacists, they say, well, these are not 21-minute clinics, these are minute clinics. So I don't know, Megan, how short is your infusion now?
So for the infusion, they're asked to be observed for about an hour. So it's about an hour, and we're talking to the FDA, to bring that number down so that across companies and across antibodies, hopefully it would be similar. For the subcutaneous injection, it's about 5 to 10 minutes to do the injections.
We're currently doing 15-minute infusions with the dual antibody, and think we can bring that down considerably, actually. I mean, the safety of these, as has already been said, is really pretty great. Very, very few infusion reactions. I think that this is something we could really, we can get in quickly.
Yeah, what Heather said, what Megan said was really important. Practicing physicians are going to need, been so kind of imbibed with the 630 news of monoclonal antibodies directed against host defenses, which have a lot of side effects. But these monoclonals directed against a pathogen prove very, very safe. And that's going to be a kind of change in thinking, I think, Rick. Thanks.
These are all human antibodies as well. I mean, that's another major advantage that they don't have, they're not chimeric animal antibodies.
Great. So thank you, everyone. Before we move on, I'm going to take one question that came in late for Mark Mulligan. So time for Mark to wake up. From Lilia from Life For Me Positive in Russia. But do you expect to see similar results if you were to use Sputnik vaccine?
Well, thank you, Lilia. That's an interesting question. That's a different kind of regimen with a heterologous prime boost. In other words, using one type of adenovirus followed by a different type. And to get the full effect of a vaccine like that, it may be necessary to have both doses, even if you're primed. So I would want to hold my judgment and sort of do the experiment before I guessed. I just am not sure.
All right. So thank you, everyone. That's great. And now I want to thank all the speakers and all the great questions from the press. And now I'll turn it over to Sharon Hillier. Thank you.
Hey, thanks, everyone. And that was some really exciting information and fascinating to listen to all your questions and the great answers. So we're going to move now first to a discussion of a new antiretroviral drug that people are pretty excited about, lenacapavir. And I'm going to call on Sarana Segal-Mar from New York Presbyterian, who's going to do the first presentation.
Good afternoon, everybody. It is my pleasure to summarize the findings from the Capella trial, a phase 2-3 global multi-center study designed to evaluate Gilead's investigational long-acting capsid inhibitor lenacapavir in heavily treatment-experienced people living with HIV with multi-drug resistance. To take a step back and provide some context to the study findings, Although there have been significant advances in antiretroviral therapy that have enabled many people living with HIV to achieve viral suppression if highly adherent to medications, we certainly have many patients who are living with HIV who received prior antiretroviral treatments who may have multi-drug resistance, leading to limited treatment options, who face a critical unmet need for new treatments. Long-acting treatments like lenacapavir with a novel mechanism of action have the potential to transform HIV care for these very patients. Lenacapavir acts at several stages of the viral life cycle, is active against HIV that's resistant to current available HIV medications, has a long half-life that allows infrequent dosing, and potentially reduces pill burden. So now for the Capella study results. We enrolled heavily treatment-experienced people living with HIV with multi-drug resistant virus who received lenacapavir subcutaneous injections every 6 months. The results demonstrate lenacapavir's potent antiviral activity when added to a failing regimen during the oral monotherapy period. The study met the primary endpoint by showing that more participants on lenacapavir achieved a clinically meaningful viral load decline compared to placebo during this functional 14-day monotherapy period, 88% versus only 17%, achieving more than half a log copy per ml drop. More importantly, lenacapavir exceeded this minimum threshold by a large margin, achieving an almost 2-log decline in viral load in 14 days, which being a clinician and researcher for many, many years, I could tell you is very meaningful and very significant by any standard. In addition, lenacapavir led to high rates of virologic suppression through week 26 when combined with an optimized background regimen, with 73% of participants achieving an undetectable viral load. Now, achieving an undetectable viral load in this heavily treatment-experienced population is remarkable and incredibly encouraging, and really supports the ongoing evaluation of lenacapavir for treatment and prevention of HIV. On the safety side, in this study, we saw that lenacapavir was generally well tolerated, no adverse events leading to its discontinuation. So in summary, the Capella results show lenacapavir's potential value as an important long-acting option that can be administered subcutaneously every 6 months as part of a complete treatment regimen to achieve viral suppression and overcome resistance for many people in this very difficult to treat population. Thank you so much for inviting me to be part of this conference.
Thank you so much. That was so interesting to see this first subcutaneous treatment option and these really promising results. I'd next like to call on April Pettit, who's going to update us on a treatment trial for pulmonary tuberculosis in people living with HIV. April?
Thank you. Thank you for the opportunity to participate. So as the background for this study, current international guidelines recommend 6 months of treatment for drug-susceptible pulmonary TB, and shorter regimens would help with treatment adherence and completion of treatment, as well as potentially decreasing drug-related adverse events and costs. And there have been phase 2 studies and preclinical studies that have shown promise for 2 agents that are being repurposed here, rifapentine and moxifloxacin. So this was a very large trial. It was conducted by the Tuberculosis Trials Consortium, which is funded by the U.S. Centers for Disease Control and Prevention, as well as the AIDS Clinical Trials Group, funded by the National Institutes of Health. The study took place at 34 sites across 4 continents, so a very large study. And the results boil down to that the 4-month regimen that had 2 drug substitutions, rifapentine for rifampin and moxifloxacin for ethambutol, was not inferior to the standard 4-month control arm. The other 4-month arm, in which there was only a single drug substitution of rifapentine for rifampin, was not non-inferior to the 6-month control, and both the 4-month control arms were safe compared to the 6-month control arm. So this rifapentine moxifloxacin arm really represents a major milestone in the pursuit of shorter treatment regimen for people with.
Okay, thank you. Our third, but certainly not least, and Rafi, you've been very patient waiting to do your presentation on the lab analysis of HIV infections in people who were participating in the HPTN 083 study of injectable cabotegravir, and what we're seeing with those resistant, whether or not they're resistant mutations. Rafi?
Thank you, and thanks a lot for the chance to present an overview of these results from our presentation earlier today. Just to remind people, these data really pick up on the story of the primary results of HPTN 083 that we originally presented at AIDS 2020 last July, when we reported the initial findings of the study, which, just to remind you, is a phase 2b3 randomized active controlled comparison of long-acting injectable cabotegravir administered at 8-week intervals and daily oral TDF-FTC for HIV PrEP among cisgender men and transgender women who have sex with men. At that time, we reported in that study, which we conducted at 43 sites in 7 countries, that we had enrolled approximately 4,500 participants, and that cabotegravir was superior to daily oral TDF-FTC for HIV PrEP in that population, with a 66% reduction in HIV incidence in participants who were randomized to the cabotegravir-based intervention. Picking up where that presentation left off, we now have completed central laboratory additional testing to further characterize the time points of first HIV infection, the pharmacology and virology of the identified seroconverters in both arms. And we had a number of key findings that I presented this morning. We had previously observed in TDF-FTC PrEP trials that the majority of TDF-FTC breakthrough infections were attributable to non-adherence. However, when TDF-FTC breakthrough did occur with intermittent use, diagnostic assays, including viral loads, were delayed or suppressed, often delaying the identification of new HIV cases. Cabotegravir and long-acting injectable cabotegravir in particular appears to also delay conventional diagnostics, and we had to expand from our original plans, our diagnostic testing and scope, to best characterize the first detectable evidence of HIV infection. So that's one point. This resulted in a reevaluation of the timing of first HIV detection in 2 previously identified infections, establishing that one of the 2 in the cabotegravir arm was actually present prior to the administration of any PrEP study products in the study. We were additionally able to identify one new baseline HIV infection in the cabotegravir arm that also existed before the initiation of any PrEP products in the study. This resulted in some minor updates to the primary results, but no change in the overall interpretation. Then we carefully looked at the pharmacology, and what we found was the following. In the cabotegravir arm, we found that injectable cabotegravir concentrations were overwhelmingly as expected with both oral and injectable dosing. In the TDF-FTC arm, what we discovered a little bit as expected was that 37 of the 39 incident infections in that arm were explained by poor or non-adherence to TDF-FTC, leaving 2 cases of incident HIV that were not clearly explained by non-adherence in that arm. With the reclassification of the cabotegravir cases that I mentioned based on the earliest detection of infection, we carefully examined the pharmacology of what is now 4 cases of incident HIV infection that occurred despite on-time injections of cabotegravir LA. Of those were first detected, all of these were actually first detected at expected concentrations of cabotegravir in blood plasma, and 2 of these 4 developed integrase inhibitor resistance associated mutations. The other 2 of those 4 did not have samples with sufficient levels of virus for genotypic testing before they were started on fully suppressive antiretroviral therapy. Additionally, 2 of the participants who we found had acquired HIV during the oral lead-in phase, both of whom, due to delays in detection of the HIV infection on the site-based testing algorithm, went on to receive long-acting cabotegravir injections, also developed integrase inhibitor resistant mutations. 1 of the 4 baseline or prevalent HIV infections whose virus were exposed to cabotegravir also developed integrase inhibitor resistance. However, importantly, despite a tremendous amount of interest and concern that infection during the tail phase would lead to integrase inhibitor resistance, based on 3 new infections that were detected and seem to have been acquired during the tail phase, none of those developed integrase inhibitor resistance. Certainly, larger data sets will be need to definitively confirm that those findings, but certainly that was reassuring. So our conclusions from the data that I presented this morning were that in the setting of an overall superior result for cabotegravir compared to TDF-FTC for HIV PrEP in the 083 population, the majority of the TDF-FTC PrEP failures was poor or non-adherence. That's quite clear. The explanation for the PrEP breakthroughs in the CAB cases, while rare events, requires additional investigation. Delayed HIV identification using currently available testing algorithms suggests the need to rapidly scale up efforts to develop new, more sensitive, point-of-care, scalable, inexpensive diagnostics. With the anticipated rollout of CAB and the promising findings for islatravir and lenacapavir in the respective stages of development. CAB PrEP failures or breakthroughs, particularly if delayed in diagnosis, may be associated with integrase inhibitor resistance that could limit first-line treatment options, both in well-resourced and resource-limited settings. But overall, the findings suggest really impressive prophylactic efficacy of both products. Thanks so much.
Thanks, Rafi. You turned your 10-minute talk into a science spotlight. So I'd like to go to, we have a couple questions and both for Saranya. If you could, one from Liz Heileman from AIDS-MAP, and she's saying, as a potential twice-yearly antiretroviral drug, what long-acting agents can you put with it? What could it be paired with? And we have a very similar question from Heather Berner from Medscape, who's said she talked to another clinician who said that they were most excited about putting this potentially together with islatravir. So the real question is, what do you put with a long-acting agent? Or do we really use these one at a time in a long-acting way, given the complexities of different companies working with one another?
Wow, I sure hope we set aside at least 30 minutes for this answer, but I don't think so. I think I will be unplugged before I'm allowed to do that. I think both of your questions are exactly on point. We are all, as clinicians and researchers, incredibly excited about this new era in HIV care. I've been in HIV care since the mid-'80s. And having seen the progress and now being able to enter into long-acting agents and especially like lenacapavir, something with its unique mechanism of action, with its unique resistance profile, and that it remains active against our currently available classes, as well as no pre-existing resistance. And its very long half-life that allows for subcutaneous injections every 6 months. It is just incredibly exciting. And I think what has really energized me as being part of this study and being a researcher and being able to present the data is the general safety and tolerability with which our participants have engaged with lenacapavir, its good safety profile, and again, the few injection site reactions and all less than grade 1 or so. So it's incredibly exciting. I think as we go forward, there will probably be more studies that look at various combinations. But I think to bring everybody's focus back till today, we're talking about a heavily treatment-experienced population that has either limited or no options, and to have such terrific results in this particular population is just very, very encouraging to myself as an investigator and clinician.
Yeah, thank you. And I think, how it can eventually be packaged with other drugs is a super important question, but I think we need to keep our eye on the fact this is amazing, right? It's an amazing outcome. I was blown away when you presented your data. We had one more question, if we could go back to that Heather asked, and I'd like to ask Mike Cohen and maybe Rafi to touch on this one, which is she said, well, we had these exciting presentations, but they're very early. And for example, with the islatravir implant yesterday data, it was about 30 people or so, I think. And the question is, what do you make of these small studies, and why do we get excited at this meeting about these small phase 1 studies? And so I want you, maybe Rafi, if you could take this one first. Why are we presenting the results of these studies and excited about them when they're so small and it's still so far away from the phase 3 result?
Yeah, thanks, Sharon. I think as people who have an eye on what a target product profile would be for something that would get us excited, early phase work on something that meets those sort of requirements, something that would be administered relatively infrequently, appears to have a good safety profile. There's non-human primate evidence to support its use. And there's already been some acceptability work about that kind of product being something that people want. I think we get excited about early promising results and just have to contextualize the limits of the available data at this point with measured enthusiasm. But I think that's sort of why I get excited about these early phase data at this stage.
Yeah, I was reflecting when I was looking at your question, Heather, about I think back about last July, we started to see these reports, exciting breakthrough in COVID vaccine research, and it would be a study of 20 people showing they got an antibody response, right? And this is sort of like that. You have to have that first step first before it moves forward. And some of the vaccines didn't move forward because there was an inadequate response. So I think it's a building block, and it's a small one, but pivotal, because negative results in these early phase studies can absolutely kill a product. Another question came in from Simon Collins, sort of challenging me that we still hadn't gotten the question answered about what to do with lenacapavir. And his question is, what about some of the monoclonal antibodies? Would you ever want to put, say, lenacapavir with some of Gilead's new bNAbs?
I think it's a very, very interesting question, and I would leave it to Gilead to comment on their future studies for treatment and prevention. I think there's a lot of speculation what you could pair it with and how and when, but there certainly is nothing that I can really speak to right now in terms of providing further, more concrete information.
Yeah, thanks. Rafi, are you trying to? Yes.
Yeah, thanks, Sharon. I just did want to mention that I think conceptually people are really interested in these long-acting antivirals and bNAbs together for maintenance of virologic suppression. And I know the DAIDS-funded ACTG is currently performing a study of long-acting cabotegravir and VRC07523LS, I believe it is. So we'll get some early data about that concept, obviously not those two products specifically, but just to be aware people are thinking about that in many contexts.
Thank you, Rafi.
So we do have one more question from Jeff Berry at Positively Aware, and this is for you, Sarana, again. You discussed in your presentation that there were some injection site adverse events that included nodules for several people that lasted maybe 2 or 3 months. How are you addressing that issue? Do you see it as a potential barrier? And this then sort of arches over to Rafi. Is this a barrier for CAB-LA for PrEP? So maybe to you first, Sarana.
Sure. So just to re-summarize the injection site reactions, the most common that we're seeing were swelling, erythema, and nodules. Occurred in less than half of the participants, and all, except for the nodules I'll comment about, were self-limited and disappeared within days. The nodules did persist for a period of time, as you recognize, and that is correct. But they're all grade 1, which means when you're looking at them being bothersome or disturbing to patients, they're all highly tolerable. We do have the patient's descriptions and comments, and they did vary, but all were, again, well tolerated. Nobody discontinued due to any adverse events related to the injections, and they certainly were not an obstacle to continuing on the study. What I can share as myself as being an investigator, there's certainly the injection site reactions were not of concern to our participants. So that was very encouraging because obviously now that we're transitioning, hopefully, with more long-acting agents and injectables, so again, lenacapavir being subcutaneous with Q6 months, very exciting. So now we're looking to see what are these injection site reactions. So I was personally gratified as an investigator to see that this was going to be something that is well tolerated, and we will see going forward, but certainly they were not an issue to the study participants.
Thanks. Rafi?
I would just say that, you know, in 083, we had reported last July that we had about 2% of the study participants in the cabotegravir arm who did discontinue for an injection site reaction or an injection-related adverse event. Happily, that was a very small number of people. I think it was 40-something participants, but I think it is clear that when you do engage with large numbers of participants, particularly in a prevention context, it's not going to be for everyone, but happily the overwhelming majority of the injection site reaction sounds like similar to what was described by Dr. Moore, were mild to moderate. And this discontinuation was a very rare event, even in the prevention context with a large number of people exposed.
Thanks, Rafi. Rick, you had one more question you wanted to ask.
There was one more question from Mark Mulligan that we passed over because we didn't know how much time we would have. So Mark, this is from Lynn Peterson from Trends in Medicine. And the question is really about false positive results. So how confident are you that your previously COVID infected patients clearly were previously infected, given that there are high false positive rates to some of the tests?
Okay, yeah, thanks, Rick, and thanks, Lynn. I'm very confident. These are people that were all PCR proven. We enrolled them back in March and April. You know, the vast majority of them, we've been following them. And then when the EUA vaccines came out, they went and got their vaccines. They continued in our study. So I think this, maybe except for 1 or 2 that had an antibody test, all of them were PCR proven. And those antibody tests were done in my lab, and they're not the commercial grade tests. So all of these, I think, are very well confirmed as having had prior COVID.
Well, thank you. And April, I'm sorry we didn't get any questions for you today, but keep up the good fight. We still need to keep the research moving on using these older drugs for TB. So we appreciate your work. I'd like to wrap up the conference today, and we'll welcome all of you back tomorrow, the journalists. I'd like to thank all of our speakers today, and thank you for the lively conversation. And we'll look forward to seeing the journalists tomorrow. Enjoy the rest of the meeting.
Thank you. Thanks, everyone.
Thank you.